All right. So thanks everyone. So the next speaker will be Gunnar Rage. Gunnar, yeah, I met Gunnar first at when I was still an undergraduate student, like I don't know, third or fourth semester even.
When was this?
1998 or 7?
No,
Okay.
99. I think 99. No.
9? No, I think 8.
Okay.
Yeah, okay, we're old. Now you know it. When Gunnar was still a PhD student at GMD first, and yeah, I don't know. I tried to do some student research project with Gunnar back then, which is eventually what you see now in Shogun, and lots of other stuff, of course.
It's the oldest part of the code, I think.
Yeah, the worst, I think, too. Anyway, and Gunnar then left to when he finished his PhD, he left to Australian National University.
Right.
For a few years. Where we used Shogun on some cluster. Like, so we had a big alpha cluster running there, and that was about the time when support vector machines entered Shogun, I think.
Yes.
And all the kernel framework and everything, all this abstraction. And then when Gunnar came back to Berlin and Tübingen, yes, we were always split in between Berlin and Tübingen then. As a postdoc first, and then he became a group leader at Friedrich Miescher Laboratory of the Max Planck Society in Tübingen, and he's now at the Sloan. No, what's the name?
The Sloan Kettering Institute. So.
Kettering Institute, and again the group leader in New York, and we all look very much forward to your talk.
Thank you. No, it's a great pleasure to be here, and I apologize. I'm still a bit jet lagged, so I might speak slower than normal. Okay, so I was asked to give an introduction to machine learning. I feel I'm carrying like old to Athens, so I can. That's what you say, right? So because you all know machine learning, I believe, at least to a certain extent. But I was told it's good to have a good starting basis. And there are also people, I think, in the live stream, at least 5 or something, who maybe the numbers shoot up now. Let's see. Okay, so I'm going to talk about introduction to machine learning in science and engineering. I've given a similar talk 10 years ago almost, and I do that general introduction first, and then that is in part 1, motivating examples and some background, and then I have some slides on boosting and support vector machines, which are 2 machine learning algorithms which are relatively famous. So, and then this is relatively high level, and then we can go into something more technical, and I give you the choice of different topics. I have a set of 300 slides, and we can choose maybe something, and then I can explain that, and maybe there's a little bit of time I can talk about some current work as well. Okay, so let's get started. I think you know this problem. You get a lot of spam emails, and you want to distinguish between spam emails and non-spam emails. So this is a spam email. I get a lottery and whatever, and this is a postdoc position announcement. I think 2004 is the date I got it. And the goal is to classify these emails into spam versus non-spam. So, and how? Okay, we can learn from previously classified emails. So we have a training set, we analyze the previous emails, and the application of this learned classifier is then to classify new emails, and then we save some time, we don't have to go through these. So that's one possible example. Another example is is an application in drug design. So here you have small molecules. These ones here, I'm not sure you can see them. You find a descriptor of these molecules. Well, let's first see what the chemist is doing. Right? It looks at these molecules, finds a description of these molecules. Then the chemist looks at those and then says, okay, this looks like an active compound. This looks like an inactive compound. Okay, so and what we want to do is and actually this is happening in circles. These compounds are classified by the chemist, then there's an experiment done, and then the chemist thinks about it again and comes up with a new set of chemicals which are active and inactive. So and here we can just replace the chemist by a learning machine, right? So, it's a bit dark, but another application of machine learning is face detection. For instance, or image analysis in more general. You have this image, and you would like to find all the faces in that image. So that's one possible application. So the kind of the premises for machine learning is that supervised machine learning is that you have a kind of a training set. You know what a face looks like, right? So you have examples of faces. You have examples of spam emails and non-spam emails. And these training examples we usually call x1 to xn, and the labels of these training examples are y1 to yn. And we then learn a function from x to y. Given x, we would like to predict y. So, and later we can predict the label of unseen examples. So we assume that these examples are generated from some kind of statistical process. So like we look at all the emails and there's some mechanism behind which is generating these emails, right? We assume that. And there's a relationship between the features and the labels, and that's why we can try to predict those, right? So if the x's and the y's would be independent, we couldn't do anything. So, and there's the assumption that all the unseen examples in the future, they are generated from the same or at least similar kind of statistical process. Okay, good. So let's look at one artificial example here. I've used these slides quite a while back. So let's say we would like to classify apples, natural apples versus plastic apples, right? And the data in this case is the set of these apples up there, and the natural, let's say, has a +1 and the plastic apple has a -1 as a label. And so the data is again our data set here. This is the set of apples, and we would like to learn how to classify the new apples. So we would like to learn this target function. So we have a distribution of these kind of apples, which is describing p of x, and the objective is, given a new x, predict the y. So what we need is, in order to do that, we need a kind of a set, a hypothesis class, something which we can use to, well, essentially we have to learn these kind of functions here. So, but we have to say, what are these functions, right? So how can we parameterize these functions? So we have to define a hypothesis class. So these are the set of all functions which go from, maybe, like, does anybody have a pointer by any chance? No, okay. Mouse, yeah, right. Here. So we have this hypothesis class, and this is the set of all functions which map from RD to ±1. And we also have a loss, which, given our prediction of a function h, which comes from this hypothesis class, and a label which tells us how bad it is. For instance, for classification, this could be the 0-1 loss. It's the loss is 1 if we mispredict, and the loss is 0 if we predict correctly, right? So, and the objective is we would like to minimize the true expected loss. And this is we would like to find the function out of the hypothesis class which minimizes this function here. This is the expected loss for our predictions. So the problem, of course, is that we can't do that because we only have a finite training example as training sample. This this training set here from 1 to n. And therefore what we can do is we cannot compute the generalization error, but we can compute the empirical error, and we can minimize that empirical error. And that's what many training algorithms or many machine learning algorithms do. They try to find a function which describes the training set well. And obviously, yeah, you have to do what you want to do is you want to have an algorithm which predicts well on the test set, not necessarily on the training set. And then there are certain tricks involving regularization and so on, some capacity control, so which help to find a function which also works well on the test set. Okay, so I would like to talk briefly about two different algorithms. One is boosting, and I'm going to use these apples for that, and then come to SVMs. Obviously, there's much more algorithms out there, and I can't give you a full overview of machine learning, so there's just too much out there. Okay, good. So let's say we have these apples, we have these examples here, true apples, plastic apples, and this is what we would like to do to classify. So let's say we have, what we need is, we need of each object which we would like to classify, we need features. We need a description of these, and let's say we just have two features in this case. So this is how red they are, right? This plastic apple is pretty red, and all these natural apples, they're pretty red as well. And these, and how heavy they are. So this is if they're heavy, and this is if they're light, right? So the plastic apples are lighter than the real apples. So boosting works like this. So you take one feature, like this one here, and we generate a decision stump, for instance. And we say everything on this side is a natural apple, and this on this side is a plastic apple, right? So, I mean, this is not perfect because we misclassify this apple here, and we misclassify this apple over there. So what boosting does is it introduces a weighting per example, and for those apples which are misclassified, it increases the weight. Okay, let's just do that. Okay, so that apple now has a larger weight, and this apple as well. The other ones have a smaller weight. And now you're asking which decision stumps should I use now? What's the best classification of this data set using some weighting which minimizes the training error? Okay, that's this one. So it gets this apple right, but it makes some other errors. Okay, so we have additional errors, so we increase the weight of those which have an error. Like this. Okay, so the next best classification hyperplane looks like this. And there's another one. And in the end, what you can do, you can just overlay these different classifications, and you get a decision boundary like this one, and this one classifies all of them correctly, right? Yeah, and this is the algorithm. So that's the other boost algorithm. That is from 1996. It's pretty old, and what it does is it takes the input examples, like here. It has a distribution which is initially uniform. It trains the base learner, which is the decision stump in this case. It computes, so it finds a hypothesis. It computes the weighted error, computes some weight for this one, and then it updates this example distribution. And then it outputs, this is done a few times, and at the end it produces a final hypothesis, which is a linear combination of these hypotheses. And this, what you see then here is this a decision boundary. So, and this has been done quite extensively. I've done my PhD on this topic. I've I don't know, published a few papers on this, and it has been used with decision stumps. It has been used with neural networks. It has been used with heuristic rules. So, and there are many improvements. So to this basic algorithm, there's some improvements which are important for increasing robustness. Some extensions for multi-class, something which improves the speed of the algorithm, so you need fewer iterations, and so on. And there's a few tutorials out there. There's also a whole there's a few talks I've given on boosting a while back. So if you're interested in this, you find some more material there. Are there questions for this? So, I mean, it's one. Yes, please. Oh, the stopping condition. That's a good point. The initial algorithm didn't have a stopping condition; it just had an iteration t. So, and then there was some theoretical result saying something about how large t should be in order to guarantee certain things. So there is in the newer algorithms there's a stopping condition. I mean, it turns out that what this algorithm does, it solves some kind of optimization problem. So it tries to find the best linear combination of hypotheses which classifies the data points best. So there's an optimality condition you can drive, and you could say if you find the best combination of hypotheses up to epsilon, right? Then you can stop and you can compute how far you are away. But that's not in the original algorithm. But some of that work which we did is related to that. Okay. More questions? So, good. So it turns out that essentially boosting is also doing a linear separation in some feature space. So it tries to find a combination of hypotheses, and in that hypothesis space, in this feature space, which is spanned by the hypothesis, is doing some linear separation. So, and that's what SVMs do as well. So there's a strong connection between SVMs and boosting. So again, we have two properties. Let's say now we look just at points, and we would like to classify these points. And obviously, the question is, how do we classify them? And let's just look at hyperplanes. The question is, which of these hyperplanes should we prefer? So. And the ideas of SVMs is to find the hyperplane which maximizes the margin. So you find the one which has the maximum distance to these data points, and then it maximizes the margin here, right? So, so your hyperplane is then characterized by the normal vector of the hyperplane, and yeah. So large margin separation, which is done in support vector machines, is that we find a hyperplane W times X, so W, that maximizes the margin. So the distance essentially between this and this point, according to this hyperplane, is maximized. It's maybe a strange formulation here, but it turns out that the optimal weight vector can be written as a linear combination of these examples. So it essentially doesn't matter where these points are in this space. I don't need the coordinate system, right? The hyperplane is just going to be a linear combination of the examples. So there's a reference to a demo. I had a demo, but it doesn't work on my MATLAB. I think I need a new demo. So if somebody can give me a new demo, I would be grateful. Okay. So the cool thing with the linear SVMs is that you can extend them quite easily to non-linear classifications. So in this case, you have data points, and you cannot separate them exactly using a linear hyperplane. So, but what you can do is you can take these data points and transform them into another feature space, and you just go from, let's say, 2 dimensions to 3 dimensions, and in these 3 dimensions you find a linear separation, and we do this linear separation here, and this corresponds to a nonlinear separation in the input space, right? So let me give you a more specific example. Let's say we have these data points here. We cannot separate them. We have these points in here. These are red, and the points here, which are blue. And you go from this 2-dimensional space, x1, x2, where is it here, to a 3-dimensional space, and you compute the third dimension, or the 3 dimensions, by taking the first dimension squared, then the second dimension is the product of the first 2, and the third dimension is the second dimension squared. And it turns out that these data points here in 3 dimensions look like this, right? You can do a linear separation here, and this corresponds to a separation, which is a circle or an ellipsoid here. Okay. Good. So, and it turns out that you just need to think about these kind of transformations, and these are done using kernels, right? You can define a kernel function, and these kernel functions essentially implicitly define this mapping from this space into this space. Okay, and this is, for instance, one specific kernel function here. The RBF kernel, which is very popular, which is also in Shogun, it was one of the first ones. So I think Shogun has, by now, maybe you know how many, maybe 40 or 50 different kernel functions. 60, Sören says 60 different kernel functions, which implement different mappings from this space into this space. And usually what you why do you need so many? These kernel functions are application specific. They depend on your data set, depend on the problem. When we look into sequences, we need a different kernel than if you look at images, for instance, right? And this is where your knowledge about the domain comes in, this embedding into the feature space. Okay. So I think this is since a few years, I would say, theoretically and algorithmically, it's relatively well understood how to do classification with a few classes. There's also not that much development, I would say. So if you have two classes and you would like to classify, learn to classify that, that is, let's say, well solved. And I think there was a phase, and maybe there's still a phase, of making these methods more large scale. I think Sören and I, when we started, we could maybe train SVMs on maybe 10,000 examples initially. And then we developed Shogun, and we could do then maybe 100,000 first, and then we worked on the algorithms, and then we could do 1 million, and then we worked a bit more, and then maybe I think the top which we had was 50 million or something. 50 million with a real kernel, right? So that's using some string data, some genome data set. So that is a development. I think by now people at Google or Yahoo or so they have even larger data sets, and they need even larger scale methods which are distributable and whatever. So I think this is would be a good direction for Shogun to develop, right? So for distributed computing. Then obviously you can do classification is one. Then you can do regression, where the output is not a binary class label, for instance, but a real value. You have to fit a function. Or there's also novelty detection, where you have a cloud of points and you would like to find what are the normal cases and what are kind of outliers, right? So here you have a cloud of points which look like the normal stuff, and then there's points which are lying outside. This looks easy in 2 dimensions, but maybe if you think of higher dimensions, that's less easy. So I would say the bottom line is that machine learning works well for relatively simple objects with simple properties. So like 2-class classification, regression, this is simple properties, and the objects are just vectors, like these points. This works well. So in recent research was really on large scale, on complex objects, on many classes, on more complex learning setups, and the prediction of complex properties, right? Okay, and there are of course many applications, even of these first kind of simpler techniques, like handwritten digit recognition, face or object detection, brain-computer interfacing, gene finding, drug discovery, and so on. So, and I think what I would like to do is just go a few of these through a few of those applications, and then we can go maybe to one part where you can choose a bit what else to talk about. Okay, I think this is a very old application, but it's pretty successful. It's the MNIST benchmark. It's for handwritten character recognition. This had 60,000 training examples, and the task was to classify these kind of images, which were 28 by 28 pixels, and tell and say what is the digit saying, right? And I think the initial performance of a linear classifier is like 8.4% error. This is a 10-class classification problem, right? And a nearest neighbor classifier is 2.4%, and SVM was 1.4. And there's different extensions, or there's also a neural network, which was very good, Linet 4. The boosted Linet 4, 0.4%, and translation invariant SVM is 0.56. So I think there's some extensions in the last few years where it is maybe 0.4, and I think that's pretty close to human recognition performance. So I think that's the error rate in the data set. Okay, face detection. Let's say you have a classifier which can classify images as either containing a face or not containing a face, right? So you have just this piece here, and it says, okay, face. Then obviously when you have an image like this one here, then obviously this classifier cannot directly detect the image. You need to guide it, or you need to put it through different pieces of the image, right? So this small piece here, where is it? Here. This small piece, that contains the face. But this piece here does not contain the face. So what you have to do is you have to run this classifier over the image and let it classify many, many times. And there's also a scaling issue, right? So you don't know exactly the scale of the face. It might be a big face or a small face. So you have to run it through different patches or different scales. So you do this, I think here this is a 600 by 405 image, and you run it at different scales. So you scale it down by 70%, a factor of 0.7. And then in this case you have 500 something thousand patches which you have to scan. And this is computationally expensive, and people have thought about how to do this, and maybe they come up with a very simple classifier first, which runs on all patches, and then it filters down the number of patches, and then classifies these images, these patches which are remaining, using a more complex classifier, and so on. So, and if you do this, for instance, on this image here, let's say after the first filter, there's 20% of the patches left. They're still very overlapping, and they're essentially covering the whole image. If you apply some more of these classifiers, then there's only about 1% of the patches left. If you apply even more, then there's very, very few patches left, but you still see there's some pieces which are not faces. And if you apply the whole classifier, then essentially only the patches which contain an image, contain a face, is left. So there's always the trade-off between accuracy and computational efficiency, and particularly for images, that's very important, because when you do the search, you have to classify many, many of these patches, right? So Søren and I have worked quite a bit on gene finding, and that is. And other people in the room, I guess. And the idea here is that we wanted to find genes which are encoded in the human genome. And so for those of you who don't know much about it, is here we have the DNA, and the DNA contains regions which are a gene, very simplified, and that gene contains exons and introns. And part of these exons and introns, well, actually the whole region which contains exons and introns, that is copied during the gene expression into mRNA, and then in that process from pre-mRNA to mRNA, these introns, the green stuff, that is removed here. And at the very end, a certain piece of that mRNA, that spliced mRNA, is translated into protein. Okay, that's pretty simple. And the machinery, the cellular machinery that looks at different signals on these sequences here, for instance, these boundaries here, they have specific properties. And these specific properties we can recognize, and we can use machine learning for this. So we have many examples of these boundaries. There's one type of boundary, there's another type of boundary, and there are other pieces which we can recognize. And we have used machine learning for that. And maybe the specific reason why Shogun is large scale is because we applied it to these kind of data sets, and these data sets are relatively big because a genome is, the human genome is like 3 gigabases long, so we have to apply it essentially for every nucleotide in the genome. We have to predict, we have to apply a classifier. So we have 3 billion test examples, right? And often we had like, yeah, 1 million training examples. So I don't want to go too much into detail here. There are different pieces of these signals. Okay, let's just go over that. So there are some computational challenges. I think the algorithms, they can work with, by now, I think with 100 millions of examples. Some of these algorithms are more online type, so they don't get the data all at once, but they go through the data from start to end, and maybe they forgot about the first ones. Then usually there are some model parameters which are to be tuned. There's cross-validation, and this gets computationally quite expensive. So you need compute clusters, you need job scheduling systems, and like PBS or grid engine. And I think what would be great if Shogun had some integration or some support of that. So that jobs are somehow distributed to a cluster. It's a bit hard, I know that. I think, okay, that's good. And I think initially we did most of that work in MATLAB, and I think it's I'm very grateful that people have done a lot of work in extending Shogun towards other languages, and I think by now it's Python which is most used with Shogun. A MATLAB interface still exists, and we still continue to use it. There's also R, and there's many other languages which are supported by now. Okay, good. So I think machine learning is an exciting research area, and I guess you're interested in it, and that's good. And there's a large number of present and future applications, and it has a reasonable theory which is underneath machine learning, related to statistics and optimization and obviously computer science. And there's a lot of interesting problems in there. Okay, so I think that first part. Let's see, how's the time? That first part is more or less done. How much time did I spend? Søren is gone. maybe 40 minutes, or yeah. So so I have 2 other parts. So, but I've only 1 hour. So let's see. I could, depending on your interest, I have a slightly more technical part. Which contains also some more general, some basic, very basic learning theory, which describes why machine learning or why learning algorithms generalize. Some more on kernels and the basics of kernels, and then kernels for structured data, like string kernels, kernels for graphs and images. Then about some extensions of SVMs. This is, I think, multiple kernel learning. I think Søren is going to talk about, so I skipped that. Something on structured output learning. There's hidden Markov models and label sequence learning. So this hidden Markov SVMs and semi-Markov extensions, and then there's a few case studies. So this is quite a bit of material. I guess I cannot talk about all of this, certainly. If there's any specific interests of you, I can talk about this. And I also have a third part, which is about one specific topic which we have been working on recently. So I could also talk about this. What do you think? Any specific opinions? Søren, do you have any specific opinion?
Practical part, maybe we should be quite strong in the first part. Maybe it's supposed to.
A practical part?
Yeah, maybe more into case studies or current work.
Okay, the case studies are on biology, so I'm not sure everybody is into it, but I can certainly talk about it. Okay. Any other opinions? I think I have 20 minutes left. Is that right? Yeah. Okay. Okay. Let's just go to part 3 then. So. Okay. It's a bit hard to see, but yeah. Well, so I thought I'd give a brief overview of kind of topics which we have right now in the group. So. Obviously, we have worked on machine learning, and we continue working on machine learning. This is like the core which we started off with. And for quite a while, we worked on genome and RNA processing and gene finding and so on. We also then extended this into deep sequencing algorithms, and there's some overlap with machine learning, which I couldn't display here, at statistical genetics. And more recently, we started looking into image analysis and clinical data analysis. So I'm working at a cancer hospital, and we have a lot of clinical records of patients, and yeah, we're trying to find good applications of machine learning where we can maybe predict the outcome of a treatment. Should we do it based on maybe past the history of the patient, or maybe the genome of the patient? We would like to take that and then predict something about how we should treat the patient. Okay, so and I think I have one topic here I would like to talk about is on multitask learning. So, and yeah, that essentially uses support vector machines in one specific flavor. So the idea is of multitask learning is that you don't have one classification task, but you have several classification tasks. So, and these are all kind of related classification tasks. So you have like not one training set, but you have several training sets. And essentially, so that's the normal setting is that you have one task. And now in multitask learning, we have T tasks, and we would like to learn T different functions. So there should be a T, and these tasks are related. So somehow the output of task I should be on the same example, should be more or less the same as the output of task of the function learned for task J for the same example. So the question is, how can we learn these functions simultaneously, exploiting all the data which we have? So we exploiting the relationship between these tasks. So, and yeah, and obviously we need also methods which can do this. We need to, since the data sets, we merge the data sets, we need to have even larger data sets, so we need more efficient methods to do that. So, and one technique is called graph-based multitask learning. So we assume that we have a classifier, which is a linear classifier, let's say. So f of x is equal to w transposed x plus b. And we have a relationship between the different tasks. And this relationship between the tasks is given as a graph. So one dot in this graph is a task. So, and these two tasks, for instance, the yellow one here and this red one here, these are very related, and there's an edge between these tasks. Whereas that node here, that task here, is not very related to this one here. And this graph structure gives you somehow the relationship between these tasks. Yeah, and what you can do is you try to find T classifiers, T functions, and you would like to find these T functions such that the empirical loss for these T functions is small. So we have T different training sets. We would like to have that these training set, these errors are small. So I think this should go from this goes from from 1 to n, and this contains all the data, all the examples from all data from all the tasks. So then you have a regularizer. This is the standard SVM regularizer, And then you have something which tells us how, or which tries to pull these classifiers together. So we'd like to have that the weight for the classifier which is associated with task S is similar to the weight of the classifier which is associated with task T. So we would like to have this should be close, right? And there is some maybe constant in front, or you could write a constant in front saying how much that should be close, right? And this graph here, this tells us how much we pull these together. So if they are very close to each other in the graph, then we say, okay, we put a non-zero weight here, and if they are not connected in this graph, then you don't put them together. Okay, so now we worked a bit on this and we came up with a more generalized optimization formulation. So you can write this optimization problem as this one here, where you define some specific embedding of these vectors, and where you just have one W which contains all the W's of all tasks, and then you can more or less write it as the standard optimization problem of SVMs. You can compute the dual of this one, and based on this dual optimization problem, you can come up with a more efficient algorithm to solve this. And there's a novel algorithm based on a dual coordinate descent, where we pick one variable, we optimize with respect to this variable, and then continue doing this. We have an algorithm for doing this, and I think this is also in Shogun. I think Chris. Yeah. It's a linear type algorithm, right? So, and I would like to give you one example where we use this. So, and there are more examples which are coming on this. So, let's say we do a gene finding, and we would like to annotate a genome. So, and obviously, what we learn from the human genome, for instance, also applies to a certain extent to a mouse genome or to a chimp genome or something, right? So there's a relationship between these different organisms. And this relationship is given by the life tree of life. These different animals and plants and bacteria are related to each other, and some are more related than others. And this is what this tree tells us, right? And the idea is that you can exploit this to combine all the data sets from the different organisms and come up with one kind of classifier which can classify where gene boundaries are and so on. So, and here's one example. We applied it to splice site recognition. Splice sites are these boundaries between exons and introns, which I showed you in an earlier figure. We consider windows around these splice sites. There's the consensus sequence, and we applied this here in this case to a set of 15 organisms. I think this is a fungus. These are plants. These are animals, and I think these are vertebrates, and these are mammals, and we have a human here, for instance, right? So, and here, I believe, this is rice, and this is the plant kingdom, and so on. And there are many other organisms, right? So, and essentially what we took is for every of these nodes here, we have a data set, which might be smaller in some cases, and bigger, like in case of human, there's a lot of data. So, and we wanted to find a classifier which is good for all organisms, more or less. That's one possible application, and by combining this data set, it improves quite a bit. This is what Chris has been working on. And I think many of you know Chris. He has been very active, I think, in Shogun, or quite active. Another application is, and this is not in Shogun yet, is a case of transfer learning. Often, what you have in image analysis, it's hard to get annotations of images. So here you would like to maybe predict or track something on an image. It's relatively easy to get labels for this in 2D, but it's pretty hard to do this in 3D. So it's very expensive essentially to label images in 3D because it takes a lot of manual work. It's much easier in 2D. So, and this is one application here where you would like to track objects in 2D through time. And there's an algorithm which you can train on this, and it does relatively well. And then in order to do the same in 3D, you would need to generate a new data set and do this. But what you can do is you can take your classifier which you learned on 2D data and apply it to 3D, and then just add a little bit of data from 3D, and then also get a good result on 3D. So that's maybe one other application of that. Okay, so there's certain extensions which we have developed. There's generalizations of this to multiple graphs where you don't have just one graph. You can learn the relationships between graphs. There's a multiple kernel learning on top of multitask learning. So that's multiple kernel learning, MKL, MTL. We can build trees from the task similarity matrix if you don't know the tree. And we have also started to apply using these kind of techniques also for clinical data analysis. For instance, when you look into different cancer types, these cancer types are not completely orthogonal. They have a relationship between each other, and we can learn on one cancer and we can apply it maybe to another. But if you have a little bit of data from the other cancer type, we can also combine or leverage this data between the different cancer types. That's one thing which we have tried recently. Okay, good. So with this, I think I've reached more or less my time limit. I'm happy to take any questions. I've also more material if you really want me to talk about anything specific, but maybe let's just open it for questions. Yeah, yes. Thank you.
I'm curious, have you also worked on L1 regularized things that essentially enforce the sparse selection of features?
For which one? The
For the multitasking learning. Or are you working on this?
I mean, the multiple kernel learning, there you would have a linear combination of kernels, which there is one version which is L1, right? So, but it's not. So there is one version where we have, for instance, let's say you have this tree here, and there's one specific version where we have okay. So what you can do is, let's say you know this tree and you would like to know what are the weights between here, right? So, and you can actually formulate this as a combination of kernels. You can formulate it as a multiple kernel learning problem by having different subparts of that tree, 0 or non-zero. So, and then you can also learn in a sparse manner these connections. So in a sense, yes. So by multiple kernel learning, which can be done with L1. So let's say you have a tree which only contains this, right? Like a graph which only describes this part. Then you essentially learn the connection between this and the rest. And you can essentially reduce learning this connection here, how strong this connection should be, to a multiple kernel learning between this and the rest of the tree. And you can do this in one go.
Your L1 is, you're enforcing sparse connections?
It's an L1 on the kernel weights, but the kernels are on these graphs more or less, right? So we have these graphs. So we do this here not with one graph. Yes. Maybe you can repeat your question.
So I was wondering about the sparsity. Is it on the edges or on the graph, or is it on the features?
It's on the graph. So let's say we have not one graph, but you have several of these graphs, and you would like to learn a linear combination of graphs. So somehow in here you have an additional weight for each graph. And if you design these graphs in a certain way, for instance, if you had a graph which only contains this connection, then you would learn this connection, and then you would learn the then you can do it. Yeah. So essentially, this linear combination of graphs boils down to a linear combination of some kind of kernels. And these weights can be learned, yes. So, and this can be done by L1 and L2. Typically, the L2 works better. Are there more questions? Yes, please.
So in this graph multiple task learning approach, so I understood that the graph is given as an input to the algorithm. So I wonder if there is a way to learn the structure of the graph from the data, and if there will be advantage doing that thing.
Yes. So maybe two things. So the graph is maybe given as is more quantitative information, right? So maybe one way is how strong are the connections between the nodes. So I mean, given that structure, there's still something which is to be learned, namely the weighting between the nodes. So this is what you can do with multiple kernel learning, for instance. So you somehow split this graph into some subgraphs, and then you learn the combination of these subgraphs in an optimal way, and this infers more or less the weighting. But to detect the structure, it's a bit harder. You can look at all combinations of graphs. Then this becomes computationally very expensive. Another way is you look just at the relationship between the tasks. Let's say you train your data on task 1, and you apply it to all the other tasks, and you see how similar, right, that task is to the other ones. So then you get a pairwise relationship between these tasks, and then you can do a kind of a clustering algorithm, let's say a single linkage or clustering, and then you get a graph or a tree out of it as well. So that's one thing. And actually, if we do this and apply it to this tree here, you get this tree back. So we just take the data from these nodes, we apply, for instance, the human data and apply it to mouse. It's closest, right? If we apply it to fungi, it is not. I mean, it doesn't perform that well. Therefore, we know this one and this one is far away. And by looking at all the relationships between each other, you can generate this graph, and it very well matches what is known from the tree of life. Mm-hmm. Other questions? Yes.
Did you use the weighted degree with shift on each genome of this organism? But somehow shouldn't you reduce your space because this genome is so big? So you use some kind of annotation-based approach to restrict your search space on all these genomes?
So we use the weighted degree kernel in this case. I think for this study even we had a simpler kernel. I mean, we wanted to make the point.
Okay.
Maybe.
Do you use the weighted degree for this? But do you have some way to reduce your search space on all these sequences? Because I mean, genomes are big and these are a lot of organisms. So do you use some method to restrict your sequences?
Right. So, I mean, we have used this on splice data. For splicing, there's so much data available. I mean, given the new sequencing technologies, you can get 100,000 of splice sites quite easily. So maybe in that sense, we have so much data for each of these organisms that predicting splicing is maybe less interesting by now. But there are other signals which are harder. Like a transcription start site is still not as well annotated, for instance. So. So we do use the weighted degree kernel, but we use efficient implementations of that. And I think Søren has worked on very efficient embeddings using hashing and everything, so into a slightly lower dimensional feature spaces, and then these become very efficient. I think how long does it take to train 1 million data point SVM? I think it was in the order of minutes, right? So it's pretty good by now.
Wow.
But we
I think it was like 1 day or so on 50 million points. So yeah, I guess.
But 1 million, I think, is pretty quick now.
It's pretty fast.
Mm-hmm. Yeah.
These pictures again depict the phylogenetic graph again, so you get similar relationship after this learning step?
Yeah, we are doing this right now, but I think the cases which I've seen so far was that it matches these trees very well. So obviously, interesting cases are those where it doesn't match very well, because then the function is not.
It's
I mean, the function which is encoded on the genome seems like not exactly matching the similarity between the genomes, and maybe there's something was moved into that genome, or something co-evolved. I don't know. It could be that for some reason the fungi and the humans do something very similar, right? Even though they are not close to each other. But these are interesting cases.
Are you planning to using this technology for splice site variants or diseases where actually the splicing is defective and maybe there are some mutations that can tell you if there are some problems that derive from the sequence actually that is different? So a multiple kernel learning on different sequences that are defective in the splicing, for example.
I mean, we look into splicing and in cancer. That's yes.
Yeah, for example. Okay.
But we haven't used multiple kernel learning for this yet. So, but maybe I can talk to you about how. But we have one study where we actually take a much larger graph and look at much more signals. By now there are hundreds of genomes, and there are maybe hundreds of genomes with annotations and with raw data which we can use. So then I think combining this and come up with a very good classifier which can predict on essentially all genomes, that would be interesting. Mm-hmm. But then obviously you have like millions or 100 millions of data points which you have to treat. Okay, more questions? Sören, you have a question? No? Yes.
We are hoping that some people on the IRC who are watching the live stream
Mm-hmm.
asking something, but
So far not? No.
so far so good.
Okay, so then maybe we go on with you, or so I mean I have to say that I'm very glad that the Shogun developments have continued, right? So that this little toolbox which we started like I don't know 14 years ago is very active, and I'm very glad that you contribute to that, and there's an active community. Thank you.
All right, thanks, Gunnar.